In the introductory video, I mentioned that biology is the scientific study of life. So in this video, what I want to do is dissect what scientific means, what the process of science is, what do I mean when I say it's the scientific study of life, and then also dissect the word life. What do I actually mean when I say something is alive, which is a slightly more complicated concept to nail down than you might expect. While biology comes from the Greek roots, bios, life, and logos, reason, logic, study of, the word science comes from Latin roots, and it means to know. So science is a way of knowing about the world. It's a way of exploring the world. It's a way of understanding the world. Now, science is not the only way of knowing about the world. It's not the only way of exploring questions about life and nature and how things work, but it's unique. Every kind of way of knowing is unique. So there are other ways of knowing. Of course, there's philosophy, there's religion, there's art. Art's a great example. I love talking about art as well. You could study, say, an oak tree in an artistic way. You could do thousands upon thousands upon thousands of drawings of that oak tree, take a look at all of its leaves and its structure and how its colors change over time, and you could draw the bark and get a good idea of the texture, and you could make different art projects with the wood itself. You could carve it into different things, and that is a way of knowing about trees and a way of knowing about that organism, but it's not a very scientific approach. Still valid, just not science. So moving on from that, what makes science special? Science uses what we refer to as the scientific method, which is a cyclical, self-correcting, knowledge-acquiring process that is based on the objective formulation and testing of hypotheses. Let's analyze that definition a little bit more. Cyclical means it goes round and round and round. The formal scientific method kind of looks like a flowchart, but it's a flowchart that circles back on itself. The arrows at the end go back to earlier steps, so you can keep doing experiments over and over again. Self-correcting, meaning that every time we go through that loop, every time we go through the process, we make our ideas a little bit better, we correct our experimental setup, we get better and better and better at this process, and we get closer to understanding whatever body of information it is we're trying to uncover. Knowledge acquiring, that's pretty self-explanatory. Every time we do an experiment, we learn a little bit something about the universe around us that is based on the objective formulation and testing of hypotheses. Now, the simple definition for hypothesis is it's a scientific guess. It's our best idea about how something might work. However, not all hypotheses are created equal. A hypothesis is a good hypothesis if it can, in principle, be disproven. We always think about scientists as trying to prove their ideas to be correct, but actually what usually happens is that we set up experiments in the attempt to disprove our best idea about the world. And if time after time after time, everything we throw at an idea fails to disprove it, if it stands up to that much rigorous testing, our confidence in that idea increases gradually with every new experiment. So that's a hypothesis. It's something that could be disproven if it is in fact false. Let's expand on this idea a little bit more. So if you can prove something to be true, it's called verifiable. So some ideas are not verifiable. You can't prove it no matter how much testing you do. So if I say, every single time I let go of this marker, it is going to fall, that is an unverifiable idea. There's no experiment that I could run that would demonstrate conclusively that every time I let go of the marker, it will fall. Even if it falls on 100% of my experiments, it is still possible that on the next one, that it is going to fly up into the air, or it could hover in place. However, that hypothesis is falsifiable, because if I do even one experiment where I let go of that marker and it stands completely still, it just floats right where I let go of it, I have disproven the idea that every time I let go of the marker, it will fall. One experimental result, the whole hypothesis comes crashing down. So that's an unverifiable idea, but it is a falsifiable idea. Now, some ideas are unfalsifiable. For example, ghosts exist somewhere, but they can choose not to be detected. That's an unfalsifiable idea, because no matter what kind of scientific equipment I bring in to try to demonstrate that ghosts don't exist, this idea already has an explanation for that. They can choose not to be detected. So there is no experiment that could be done that would say, nope, ghosts simply do not exist. It's an unfalsifiable idea. If an idea is unfalsifiable, it might still be true, but it is not science. It can't be a tested hypothesis, so it's outside of the realm of science. Science confines itself to testable hypotheses, and a testable hypothesis has to be falsifiable. In other words, it has to be a natural explanation for some observed phenomena. Now, some things are unverifiable and unfalsifiable. So there's an idea in philosophy called last Thursdayism. It goes like this. The universe came into existence for the first time last Thursday. Prior to last Thursday, every little bit of energy in the universe, all of the matter that we perceive, it was all diffused all over the place, like one big cloud, a big nebula of nothing. And it just sort of all randomly, by random chance, came together into its current form last Thursday. Now, I know what you're thinking. I have memories from before last Thursday. But those memories are a byproduct of the structure of your brain and the way that your neurons are connected together, and that is part of the universe. So the idea that the entire universe came into existence for the first time last Thursday includes an explanation for where your memories come from. This idea can't be tested. It's not verifiable, and it's not falsifiable. There's no experiment that we could run that would prove that the universe was here before last Thursday, because the last Thursdayism model of the universe makes exactly the same predictions as the more what we might call the standard explanation of the universe, where we do have a history that precedes last Thursday. Now, this seems like a very, very silly idea, but this does happen in science periodically, where you'll have two models of the universe, two theories, if you will, and both of them make exactly the same predictions about the universe around you. So experimentation couldn't really delineate the difference between the last Thursday model and the long history model of the universe. So what do you do in this situation? Do you just pick whichever model of the universe appeals to you more and run with that? No, not so much. In this situation in science, we appeal to something called the law of parsimony. Now, in pop culture, it's usually referred to as Occam's razor. You may have heard that before. The idea is all things being equal. So two ideas, two scientific ideas, two theories of the universe, two hypotheses make exactly the same predictions. They are equal in what kinds of predictions they make about the universe. The simpler explanation is the one that you should use while developing the rest of your ideas. It's not necessarily correct, but it's easier to work with, so it's more about pragmatism at that point. A universe that spontaneously came from nothingness last Thursday would require us to invent entirely new fields of physics that explain phenomenon that we've never actually witnessed before. So that is the more complicated model of the universe. Whereas the long history model, which assumes that the universe operates according to predictable laws and that forces that were at play in the past are the same forces that are at play in nature today, is a lot easier to work with. It requires less hand-waving of explanations. So let's take this idea a little bit further, get into some more detail. Once we have our hypothesis, we need to come up with a way to test that hypothesis. We need to design an experiment, which means we need to analyze our idea and figure out what predictions about the universe our idea would make. In order to do this, we use a form of logic called deduction. Deduction is logical statements that take the form of if this, then this. If A, then it follows that B will occur. So if this hypothesis is true, then this is the prediction that we would expect. I'll give you an idea. Let's say that we take a look at DNA evidence. We started noticing when we did DNA testing that closely related individuals had more similar DNA to each other than distantly related individuals. Like, there's no organism on the planet with more similar DNA to mine than my brother, because we share a very recent common ancestor, our parents. Taking that idea that closely related organisms are more similar genetically than distantly related organisms, we could take a look at some of the ideas that Charles Darwin wrote down in his book, The Descent of Man, which was kind of the sequel to On the Origin of Species. In there, he speculated that humans are a part of the great ape family. They're related to gorillas and chimpanzees and bonobos and orangutans. Now, we can take that idea. We can say, if closely related organisms have similar DNA to each other, and humans and other great apes are closely related, then it follows that if we were to do a DNA test, we would find that humans and chimpanzees and gorillas are more closely related to each other than they would be to, say, a bat or an iguana or any other kind of organism you care to name. Now, Darwin wrote these ideas down way before we had genetic technology. So before we did this test, we didn't know what the result was going to be. It was not a given that it would support his idea. But when we did the test, we found that, say, humans and chimpanzees are profoundly related to each other. Over 98% genetically identical, as a matter of fact. So this test supported that idea. So now that we've talked a little bit about the nature of science and what a hypothesis is, let's take a little bit more of a look at the scientific method. And you might be surprised to learn that the scientific method is probably something that you use in your life just about every single day. You just don't think about it in the step-by-step process that the scientific method is kind of designed to be. So take a look at this slide here. One thing you might want to notice on this slide is that there's a little pulsating question mark icon on the top left of the slide. Whenever you see one of those in one of my presentations, it signifies that there's a question that I'm going to ask you. Okay, so if I just click that here, whenever the symbol appears on the slides, a question will be posed to the class, and I'll usually tell you when you should pause the video to try to figure it out for yourself. The scientific method starts with an observation. So let's say you're on your way to biology class, you're excited, it's your first day of biology class, you're ready to go, you get in your car, you turn the key, nothing happens. The engine doesn't turn over, absolutely nothing. So your observation is, my car won't start. Now, that is going to pique your interest, so you'll probably ask yourself a question, like, why won't my car start? What are all of the possible reasons that a car would not start, and which of these reasons are testable? It could be that there's gremlins in my engine, but since I don't have a theory of gremlins to guide my thinking on that, let's go with some of the more testable ideas. So here's the question for you. Go ahead and pause the video and just think up 2 or 3 different reasons why your car might not start. You'll probably have one that pops to your head immediately, but think of a few other alternatives, because often this is what we have to do is, if we can't find support for one idea, we have to, quickly move on to some other idea. Science waits for no one. So give the video a quick pause, write down 2 or 3 ideas that could be tested. All right, so you got your ideas, I got my idea. We've already made the observation the car won't start, and now we've asked a question, why won't it start, and we've generated hypotheses. You got a couple of them there. I'm going to go with my first hypothesis, which is my battery is dead. My battery is dead, and that's why my car won't start. Now, my battery is dead is a good hypothesis because it makes predictions which could be tested. What kind of predictions does my battery is dead make? Well, it makes a prediction about what would happen if I ran the experiment of turning my headlights on. If the battery is dead and I flip my headlights on, then they shouldn't actually turn on. I shouldn't see any light coming out. However, if the battery has full charge and there's some different problem wrong with my car, then when I turn the headlights on, the headlights should come on even though the car itself will not start. So this is an idea that I could disprove. If the headlights come on, it's not the battery. I would have disproven that idea. So that's made a prediction, and then it also kind of implies the experiment. The experiment is just to turn on the headlights and see what happens. Now, based on the result of that experiment, we're going to do something else. So, if the experiment does not support the hypothesis, in this case, that would mean that I turned the headlights on and they came on. So the battery is fine. I've disproven my hypothesis. I have to go back and write a new hypothesis. That's what's supposed to happen. Usually what happens is that we go back and we modify our hypothesis. We tweak it to be consistent with the experimental results, and then we make new predictions based off that modified hypothesis, and we test again and again and again. But we need to do some reworking at the hypothesis step if the experiment doesn't support it. If the experiment does support the hypothesis, then that's great, but I haven't actually proven anything. So I turn the knob and the headlights do not come on. That supports my hypothesis about the battery. And then I think, next experiment is if I jumpstart my car, that is going to get the car started. That'll charge up the battery and that will solve the problem. So that's a new prediction for my hypothesis, and it implies a new experiment to jumpstart the car and see what happens. And I try it and nothing. I still can't get the car to turn on. Because it turns out it wasn't that the battery was dead. Maybe instead it was that the battery cables had been frayed. They'd been gnawed through by an animal in the middle of the night, or they just came loose for a different reason. Just because an experiment supports a hypothesis once doesn't necessarily mean you've proven that hypothesis true. There's always more and more testing to be done. So this is the general, super simplified scientific method. Observation, question, hypothesis, prediction, experimentation, and then based on the results of that experiment, you go back to one of the earlier steps. Now, there is no one scientific process. Every single scientific team is going to develop their own approach to science, which will be appropriate for the kind of tests that they do. An ecologist's approach to the scientific method is going to be extremely different from a biochemist's approach to the scientific method. So there is no one method. This is just kind of a general guideline. It's usually the kind of thing you see in a textbook. One feature that our experiment with the headlights was lacking that any good scientific test should have was a control group. Kind of hard to have one when you're just trying to get your way to biology class, but if you are running a scientific experiment, you have some test subjects that you're running it on. Let's say we're running an experiment on mice. And we are trying to test what would happen to the mice if we change variable A or change variable B. And these could be anything. Changes to their diet, giving them some medication, infecting them with some material, or just changing the temperature in their cage. What would happen? Our results from those experiments will have no value whatsoever unless we have something to compare them to. And that's what we call a control group. A control group is a set of subjects that does not receive the specific factor that we are testing for. But in all other ways, they should be as identical to the experimental group as they can possibly be. So if we were testing a new kind of diet, we want to know if this diet will cause you to gain weight, and we get a bunch of mice and we feed them this diet and they all gain weight. Have we proven our hypothesis? Well, no, because maybe this is a breed of mice that is disproportionately likely to become obese over time. Or maybe they became overweight for a different reason. Maybe there wasn't enough exercise available. We don't know that the specific thing, the specific diet that we were giving them, is actually the factor that gave us that result. What we need is another population of mice that's identical to the original population that doesn't receive that diet, that just gets normal, say, lab blocks, what mice usually eat. And then we can compare how much weight was gained by the experimental group versus the control group. So let's look at a real example of the scientific process in action. Alexander Fleming made a profound observation. He noticed that the bacteria that he was trying to grow in his lab would not grow on a petri dish around the locations where a particular kind of mold was also growing. So he speculated that that fungus might be emitting some kind of chemical that was inhibiting the growth of that bacterium. And he isolated the chemical and he calls it penicillin. You guys are probably familiar with penicillin. Now, just because a effect can be observed in a petri dish does not mean that that same effect will occur in a living organism. So we had to do more testing. So the observation is that bacteria seem to not grow when this chemical is around them. Flory and Chain, another group of scientists, asked the question, can this chemical, penicillin, produced by the Penicillium notatum fungus, be used to fight bacterial infections? Now, their hypothesis is that the chemical penicillin is interfering with and slowing the growth of hemolytic streptococci, the kind of flesh-eating bacteria that was of interest here. It's related to the bacteria that give you strep throat, and this sort of thing. So their prediction is, if the chemical is actually having that effect, that it might still have that effect inside of a living organism. So mice treated with penicillin will be less likely to die from bacterial infections than a control group. So the experimental group are the mice receiving the penicillin treatment. The control group are the mice that do not receive the penicillin treatment. Now, in order to test whether or not they are likely to become infected with this bacteria, we have to expose both groups to the bacteria, and then whether or not they had penicillin should be the only thing that's different between the two groups. So we have our question, our hypothesis, our prediction. The experiment is to take 8 mice and infect them with H. streptococci. 4 of the mice are treated with penicillin. 4 of the mice are our control group. They are not treated with penicillin. Our results of this experiment are that the untreated mice, the control group, died within 17 hours, but every single one of the 4 mice in the experimental group survived. Now, that's a good result, and it's a result that is significant enough to indicate that we should do more testing in this area. But you can't publish a new drug on the results of an experiment that only involved 8 mice. Maybe, just by coincidence, you happen to pick 4 mice for your experimental group that were just disproportionately resistant to this strain of bacteria. So you have to repeat that experiment with a larger sample size. So we did the experiment again with n equals 50. The number of mice was 50. So you have 25 getting penicillin and 25 not getting penicillin. And again, every single mouse that got the penicillin survived. All of the mice that did not receive penicillin died. So this is the beginning of the journey to getting actual antibiotics. Antibiotics are chemicals that fight bacterial infections, and now we have lots of different antibiotics, and it's a good thing because the bacteria are developing new resistances all the time. So we are constantly doing experiments like this, trying to find new types of drugs that we can use. One last note on the nature of science is the term theory. Now, in everyday conversation, if you look up the definition of theory in the dictionary, you will find that it is a guess, right? And that's how we usually use the term theory. But if you take a look just a couple of entries down in that dictionary passage, you will find another definition for the word theory. And this is the one that scientists are using when we use the term theory. A theory is a broad explanation for many different phenomena, and it is supported by a large body of evidence. That is, we've done experiments based on this theory over and over and over again, and just about every time we have found support for this theory. That the grand mountain of evidence that we have vastly supports this theory as opposed to alternate explanations. When a scientist says theory, they do not mean a guess. They mean a model of the universe that they use in order to understand observations that they are making. It's the framework from which they make new hypotheses. So here's some ideas of theories that you may have heard of. Germ theory, the germ theory of disease, the idea that bacteria and viruses make us sick. That is a scientific theory in biology. General relativity, the idea that gravity and massive objects alter the fabric of space. If that was not an accurate theory, then our satellites and our GPS units wouldn't work because they are based on those principles. So it's a very accurate theory, but it is a theory nonetheless. Atomic theory, the idea that atoms exist. Atoms are the little particles that make up chemicals. These are all theories, models of the universe that we use for explanations. And evolution is the unifying theory of biology. It is the only explanation that we have that explains all of the different phenomena that we see. So you'll notice me going back to evolution periodically during our discussion, and we'll spend an entire unit essentially just talking about evolution later on.